The line between myth and biology is thinner than a strand of hair. It's a line we walk every day here at EnfinityCast, peering into the shadows where science fears to tread. I'm your host, Vesper Thorne. Tonight, we're not just telling stories, we're decoding them. We're discussing monster genetics, analyzing the biological fragments left behind by creatures that shouldn't exist. And to help me navigate the double helix of the unknown, I'm joined by the man who runs the sequencer himself, my co-host, Elion Quanta.
It's good to be here, Vesper. Though after the week I've had in the lab, I'm not sure good is the right word. Some things, once you see them in the data, you can't unsee.
That sounds ominous. Let's start with the sample that came in from the Pacific Northwest, the one labeled Patterson-Gimlin site, Sample 7, the alleged Bigfoot hair.
Right, so we ran it through our most sensitive next-generation sequencing. At first, the results were disappointing. 98% of the mitochondrial DNA matched Ursus americanus, a black bear. Case closed, right?
But that's not what you're implying. What was in the other 2%?
That's the problem, Vesper. It wasn't contamination. The nuclear DNA, it was a mosaic. Part bear, yes, but stitched together with sequences that were distinctly primate, and not any known primate. We found genetic markers that were closer to hominid lineage, but older. And one sequence, a huge chunk of non-coding DNA that has no match in any database on Earth. None.
No match at all. So it's not a bear. It's not a man. It's something else. Something that shares a forest with one and a bloodline with the other.
It's an anomaly that walks on two legs. And that's not even the strangest thing we analyzed this week. Let's talk about the Texas blue dog, the alleged Chupacabra.
The desiccated canine remains found on that ranch outside of Cuero. The official report said it was just a coyote with severe sarcoptic mange.
And genetically, it is mostly coyote, Canis latrans. But we focused on the genes responsible for hair growth and skin development, the ones mange would affect. And we found a mutation I've never seen. It wasn't a degradation of the gene. It was an entirely different, highly efficient gene for regulating body temperature through blood consumption.
Wait, you're saying its DNA was coded to be a blood drinker?
I'm saying it had a unique adaptation for processing hemoglobin as a primary supplement to its diet, far beyond what you'd see in a tick or mosquito. It's a predatory adaptation that doesn't belong in a canine. It's as if something took the blueprint of a coyote and, rewrote a few terrifying paragraphs.
So we have a primate that isn't a primate, and a coyote that isn't a coyote. The evidence doesn't lie, Elion. It just whispers truths we aren't ready for.
And Vesper, that reminds me, the new sample just cleared quarantine, the one dredged from the lake in Scandinavia, the gelatinous membrane.
We'll have to save that for next time. We're out of time for tonight. The question remains for our listeners. When you map the genome of a monster, do you rob it of its mystery? Or do you simply give the terror a name?